to be here and providing an update on recent developments on the tool ProDy. ProDy stands for protein dynamics, but as you're going to see, it's actually much beyond protein dynamics. You can use it for analyzing large systems, complexes of proteins with DNA, RNA, or other molecules, the lipids. But originally it started as a tool for protein dynamics. Now, I would like to just introduce to you the interface, ProDy, that we have. The purpose of this workshop is to let you familiarize with this tool. I will give some general backgrounds in the next 50 minutes, followed by questions and answers. And then James Krieger and Anupam Banerjee are going to give are going to run the hands-on session to let you do applications. So I think it will be very important to follow both parts in coordination. Now, in addition to ProDy, we have also another interface called Dynamics. Both of them originally started with elastic network models, but we have extended well beyond that, including hybrid models that combine with molecular dynamic simulations. The major difference is that Dynamics has been designed for biologists and doesn't require any programming language. ProDy, on the contrary, is designed for computational biologists and would be requiring a Python programming language. So there's a little bit of more effort to use ProDy. On the other hand, it is worth it because you can generate lots of data, make lots of predictions with little effort for learning the language. And there are excellent tutorials that are going to guide you. You can even learn by yourself, but here we're going to make it easier for you. Now, for your information, it has been downloaded actually, this server, the program, more than 2 million times since its inception in 2011. So that is, and it has been used by many countries, as here you can see. United Kingdom it turns out to be in number 4 after United States, India, and China, and before Germany, Japan, and France. So we have also, I would like to emphasize how, I don't know why it's not working. We have the number of downloads. Originally, the first year it was only 8,000. The next year was 35,000, and so far it is again more than 2 million. And the number of unique users is more than 140,000. So it's a broadly used interface, and hopefully you will be using it from now on your research. Again, the foundation is we use elastic network models. Why do we use network models? This is a simplification. It makes the structure, visualization, and analysis much easier, much more efficient computationally. And it provides them a major advantage of considering the structure as a whole, not a certain component of it. So in this regard, it is very good for large systems, collective motions, long time processes, which you cannot do with full atomic simulations. Elastic network models, or network models in general, incorporate structural data, and you could incorporate that at multiple levels of resolution. And we have been also taking advantage of theories that from other disciplines like polymer physics, graph theory, etc. Now, the major point is that you have, for example, in the protein data bank, lots of structures, but proteins are not fixed structures. They are not static. They move, breathe, work, dance, interact with each other all the time. and that's the way they achieve their function. So it is possible to examine, for example, local motions at the interface between different domains, different subunits, or global motions of the entire here chaperoning, as you can see. So it is important to understand that the major utility is to consider large systems. If you are interested in a small portion, very detailed chemical interactions, you don't use elastic network models. But if you are interested in the overall big machine, for example, the way a virus, in this case, this is a virus with its genetic material inside, the way it tends to move, or how the activation of a given subunit by ATP binding takes place, the ProDy is very useful. Elastic network models are very useful. So if you would like to see the big picture, here are different length scales and different time scales. So at this level, femtoseconds, you have bond vibrations, which are less than certainly nanoseconds or even angstroms. But as you go to higher scales, both in space and in time, you have different sizes of motions that come into play. We are very much interested in that region where you have multimers or machines, molecular machines, and the way they use. And that's exactly where elastic network models are important for bridging between structure and function. Is there a question? So, for example, this static image we turn into a movie. That's what we do.
Sometimes that can be either.
I'm sorry, there is somebody asking a question. Okay, I don't know. Or somebody needs to turn off their microphone. And you can also apply the same theory to cryo-EM density maps to make an assessment of the types of collective motions that cryo-EM resolved structures undergo. That is an application to a mammalian chaperoning here, showing how the 8 subunits would be concertedly moving to achieve the chaperoning function. Now, here is a summary of of my presentation. First, I'm going to talk about two theory elastic network model foundations. One is called the Gaussian network model, and the other is the anisotropic network model, GNM and ANM. And then I'm going to talk about what the types of analysis that we do for understanding function, generating functional information from sequence and structure. Today, we're going to talk about how you generate from structure. Now, starting with the theory, again, two elastic network models. And here are the references. The Gaussian network model originally was introduced in 1997 in this journal that doesn't even exist anymore, but this paper is still being very highly cited and accessible on the internet. And this is one of the recent upgrades. Now we have a database where then in 2016, we applied to all protein data bank structures and generated results. So you can even retrieve directly the results. But for new systems or for your models, structural models, ProDy is much more flexible. You can do lots of computations. The anisotropic network model was introduced in 2001 when I was then a professor in Istanbul, Turkey. This is the year I also moved to the United States. And then a recent version, a server for the anisotropic network model is also accessible online, which we published in 2015. Now, these are the basic, the original study and recent upgrades on those systems. ProDy incorporates all of them and also even the most recent 2021, 2022 upgrades, as you're going to see. Now, what is the basic approach? the basic approach is we take a structure and we turn it into a network model. So this is the original ribbon diagram you are familiar with. Now we turn it into an elastic network model. So if you enlarge a portion, what you're seeing is an ensemble of beads and springs. Beads usually are placed at the positions of alpha carbons, and springs connect those that are spatially close in space. They may be connected by chemical bonds or not connected, but just physically interacting. This is another way of describing, again, your protein network model. What we are essentially doing is to examine in this energy, the energy minimum that the folded state represents. The energy minimum is approximated by a smooth linear curve, linear force for a quadratic potential, and that's what elastic models mean. They obey a quadratic dependency on strain, on the extent of elongation, stretch. And then for a given system, we start to determine what the motions are. Here is a summary in this movie. Suppose you take a given PDB structure, let's say aniline kinase, focus on the backbone, identify all the alpha carbons shown here by spheres. These are the nodes of our network. And then those connect nearby, we connect by springs. Here is the network representation of adenylate kinase. And then we start to do what's called a normal mode analysis, determine the most collective motions, mode 1, mode 2, mode 3, etc. And so this is, I'm going to explain how we do that, these things, okay? Now, more details. A Gaussian network model, again, each node represents an amino acid. And so we identify their we identify the positions of amino acids with those of the alpha carbons. And then we define their position in space by Ri, as you can see here, the vector. And then we have springs connect all node pairs within a certain interaction distance, let's say 10 angstrom. And then the name Gaussian means that the nodes themselves undergo Gaussian fluctuations. Or if you wish, the internode distance, Rij, the change in internode distance, that Rij, which you can see here, this is the difference between instantaneous distance and original distance. This distance obeys also a Gaussian distribution. It's centered around 0, and it can get larger or smaller, obeying a Gaussian distribution. That happens when the prevailing potential is a harmonic potential. So that's why it's called Gaussian network. And you can represent the overall fluctuation or motion of the molecule in terms of the motions of the individual nodes as a vector, as you can see here. Originally, we were inspired by the Rouse model of polymers, where you have a linear chain, nothing connected, everything is just connected to its first neighbor. Under those circumstances, for such a linear chain, the connectivity matrix is known as the Kirchhoff matrix, where all the elements that I haven't written are 0, and there is a band of non-zero elements, such that the diagonal elements represent the degree or the coordination number of individual nodes. So that one has 1 neighbor, so it's 1. The second one has 2 neighbors, so it is 2, and it goes 2, 2, 2, 2 all the way to the end, and then 1. And then the off-diagonal terms are always equal to -1 when these 2 nodes are connected by a spring. So in this classical Bidin spring model, only the first neighbors are connected, so only the first off-diagonal arrays here. are -1 and everything else is 0. So for such a system, you can represent the overall potential as a sum of harmonic energy, harmonic functions, quadratic functions, if you like, harmonic, and which scale with the fluctuations in the distance between nodes 1 and 2, node 2 and 3, etc. And there is a force constant. And you can represent that in a concise way, as you will see, by replacing the change in the distance by the difference in the fluctuations of the individual nodes. And in this case, you can obtain the same expression by pre and post multiplying the Kirchhoff matrix by the position vector row and column. So the product, also pre-multiplied by the force constant, is going to give you the overall potential. So this is a concise way of doing that, calculating the potential. And you can write as, delta R transpose here, the Kirchhoff matrix gamma, and delta R pre-multiplied by the scalar. Now, in the case of proteins, the major difference, the same equation applies identically. The same equation. The major difference is that the Kirchhoff matrix is not any longer just a band composed of 3 rows like diagonals, but now we have off-diagonal non-zero terms equal to -1 whenever the distance between the ith and kth residues, so these are, you can think of this as the residue numbers, whenever their distance is closer than a certain cutoff distance, which is the interaction distance, then you have -1. So you have lots of elements here, which refer to amino acids that are far along the sequence, but close in space, something that we'll call tertiary contacts. It's very similar to contact maps you may have seen in the biochemistry textbooks. So once you know gamma, and you can evaluate gamma very easily knowing the PDB structure. Shortly, James and Anupam will talk on that. Now, once you know gamma, this is it. You already know a lot, because if you would like to calculate any statistical mechanical average, this is the usual formula for a continuous function. You look at the probability of X for evaluating the average X over all possible, the average f of X over all possible X values. And so you can think of the probability, if you wish, as a weight function divided with sum of the weights, which is nothing else than the partition function. This is classical statistical mechanics, right? Now, in our case, suppose we would like to calculate the cross correlation between the fluctuations of residues I and J. So delta R, delta Rj, the correlation cosine, averaged over all possible motions, over all possible changes, fluctuations of the molecule. So in this case, you have the same function here within the integral, and then the weight is nothing else than an exponential factor where V, the potential, is simply that expression, okay? So if you substitute this expression there, this is called the generalized Gaussian expression. You can take the integral, and I'm skipping the mathematical steps, but the result is a very simple equation. The result is that delta Ri, that delta Rj, the cross correlation between I and J, amino acid, the way they move in space, is simply proportional to the ijth element of the inverse, actually pseudo inverse of gamma. So that is a very important, very simple rule. Once you construct the connectivity matrix, Kirchhoff matrix, if you invert it, and actually this is a pseudo inverse because there is a zero eigenvalue, but you can calculate the pseudo inverse by eliminating the zero eigenvalue. So once you invert it, the ijth element will be scaling with the cross correlation between residues I and J. And then there is a proportionality constant Gamma, the force constant, is obviously in the denominator because the larger the force constant, the stiffer the connections, and the smaller the fluctuations, right? So gamma provides a complete description of equilibrium dynamics. In addition to cross-correlation, obviously, you would think that you can calculate, of course, the mean square fluctuations too, just by having I and j equal to each other. So that our I square is nothing else than the mean square fluctuation of raised to I. And it is proportional to the diagonal element of the pseudo-inverse. Simple applications. Now, if you would like to calculate the cross correlations of hemoglobin, here is an example. You have all the residues. By the way, hemoglobin is composed of 4 subunits, as you can see here. They look very similar, but when you start to do the calculations, you're going to see that the cross correlations you can represent by a 2-dimensional map, each element color-coded by the level of correlation. In this case, the subunit 1 and subunit 1 are very strongly correlated, as well within subunit alpha 1, you have very strong correlations within beta 1, etc. But then the cross correlations between alpha 1 and alpha 2 are very weak, whereas the cross correlation between alpha 1 and beta 1 is relatively stronger. So that is actually called a dimer of a dimer, because 2 of the monomers are very strongly coupled, and the other 2 as well. And there is an anti-correlation, which means opposite direction movements between them. So things that you would not see very clearly from the 3-dimensional structure, by calculating this type of cross correlations, you understand better the mechanisms of motions. So that is the cross correlation map. That are I, that are j. And then again, if I is equal to j, we have the diagonal elements, which we have plotted here. So what has been plotted here is the mean square fluctuations of amino acids, peak referring to the most mobile regions and minima to the most rigid regions. And actually, we're showing that only for alpha and beta subunits. We are also comparing those theoretically predicted mean square fluctuations by what is called B factors. B factors are the Debye-Waller, also called Debye-Waller factors. They are representative of the mobility of individual atoms in the crystal structures, how much each atom fluctuates. So in the PDB, in the protein data bank, in addition to the XYZ coordinates of every single atom, you have another column that lists the kind of uncertainty in those positions. So this is the B factors. So you have XYZ are the mean positions, if you like, and the B factor is some kind of a standard deviation. So this standard deviation, physically, it relates to the mean square fluctuations of the residue, under physical conditions. And even though there are many approximations, as I'm going to discuss, but B factors have been a good measure of testing the predictive ability of those network models by simply, because B factor for raised to I would be directly scaling with the mean square fluctuation of raised to I or atom I. So we have been doing that and observing always a correlation of the order of 0.7, which is very good for such a coarse-grained method. However, one needs to be careful when B factors are used for validation or calibration, because the B factors for a given protein, let's say this green protein, are also affected by crystal contacts. The way a given protein crystallizes, if you look at it, it's not a single molecule, but it's an array of molecules arranged in well-defined packing geometries. And this packing geometry may be even different under different crystallization conditions. So for the same designed peptide here, a small protein, you have 2 different crystal packing. Now, what happens is that we are interested in the mobility dynamics of the protein, but the information we extract from these crystal structures as the B factors is actually their dynamics in the crystal environment. So some regions that make crystal contacts may be artificially, here crystal contacts, may be artificially restricted because of contacts, whereas under physiological conditions, the protein itself can move freely. So that's what we observed. For example, theoretically, we observe some regions to have very large fluctuations with mean square fluctuations. But then when we look at the B factors, they are much lower. And these lower values originate from those artifacts of the experimental data. Now, if we repeat our computations using this pair of proteins or the entire protein in this crystal environment, we practically reproduce the experimental B factors. So we make the theory as if we are representing now the crystal environment. But in reality, the protein itself would undergo much larger fluctuations at these positions. So one needs to be careful. All right, and then if you have the cross correlations here, and you would like to divide by mean square fluctuations of the individual amino acid, square root of that root mean square fluctuations in a sense, this gives you then just the correlation cosine, a purely orientational correlation between amino acids I and J. Okay, and that varies from -1 to +1, -1 being when the two amino acids undergo exactly opposite sense directions, but in a very concerted way. So they are strongly correlated. They are called anti-correlated. And +1 when they move again concertedly in the same direction. Okay, fully anti-correlated, fully correlated. Now we represent the elements of the correlation functions, the cross correlations, as elements of the covariance matrix C. And the covariance matrix, by definition, since these elements were, remember, they were proportional to the inverse of the gamma matrix, the covariance matrix itself scales with the inverse of the Kirchhoff matrix. So that is, and the proportionality constant is nothing else than 3kt over gamma. So this is the way the covariance matrix looks like in the GNM. It's an n by n matrix, and it gives you information on how much the residue 1 and 1 are correlated. This is the mean square fluctuation here, and the off-diagonal terms will be giving you the cross correlation. So practically, when I showed this diagram for hemoglobin here, we are just showing the elements of the covariance matrix. That's all. Very simple. So we are just showing in color code the elements of the covariance matrix. Okay. Now, an important advantage of the elastic network models is to evaluate the normal modes of motions. And this is done by eigenvalue decomposition of the Kirchhoff matrix, where you have you can rewrite the Kirchhoff as a product of the matrix of eigenvectors, eigenvalues, a diagonal matrix, and transpose of this matrix. So that is when, and this is the matrix of eigenvectors, where you have each eigenvector written as a column, and its transpose would be each eigenvector written as a row. Now, when you do the eigenvalue decomposition, it means you are writing gamma as a sum of eigenvalues and eigenvectors. Each eigenvector represents the profile, mobility profile of amino acid in that particular mode. And the eigenvalue here is like a weight factors. It is also gives you, it is going to be, usually we are interested in the inverse actually. And when you take the inverse, you just have, you can show that this product of eigenvectors is the same, but the eigenvalue, you take the reciprocal. And if a given eigenvalue is small, it scales with the frequency of the motion of that particular mode. Smaller value means a bigger contribution to the overall covariance here. So it is because 1 over lambda k will be a big number, and so on. So we have, you can calculate how different modes contribute to the mean square fluctuation by simply replacing this inverse here by the summation over eigenvalues and eigenvectors, each of them. And then you can look at the contributions of individual modes. So we have done that, for example, here for HIV protease. When you do the slowest modes, which are those with smallest eigenvalue, they're going to give you the eigenvectors, the profile, the mode shape, it's called mode shape, is very smooth as a function of residue index. You will have minima and maxima in a more distributed way. The square fluctuations driven by the slow modes. In the case of fast modes or high frequency modes, you have peaks like that. And those peaks will refer to the regions that are most important for stability. Usually these are at the folding nuclei, and they are not going to sustain any mutations, for example, because then you perturb the stability. On the other hand, when you look at the slow modes, also called global modes, low frequency modes, the profile that you obtain for the motion of the individual residues will give you information on functional properties. So you can detect here the active site as a very, precisely, tightly defined region with minimal fluctuations, which is critically important for positioning the catalytic residues exactly to achieve function. Whereas the recognition loop is highly flexible because it is the recognition loop and it needs to accommodate substrate binding, etc. And so in summary, you have the structure. Whenever you have a given structure or a structural model, we turn it into a network model, evaluate the connectivity matrix or Kirchhoff matrix. And then from here, you can calculate the mean square fluctuation of residues or their cross correlations through the inverse of the gamma. And then you can dissect the overall dynamics into the contributions of individual modes. Usually the lowest frequency modes are going to give you information on functional properties, and the highest frequency modes are going to give you information on residues important for stability. So this is a simple recipe for you. You take the coordinates from the PDB, protein data bank, construct the gamma, the eigenvalue decompose, and then you can, the dynamics, as I said, is a superposition of all modes. All modes, each mode is represented by an eigenvalue and eigenvector. The eigenvalue scales with the frequency squared of that particular mode. The eigenvector is an n-dimensional vector, the kth element of the ith element of which represents the displacement of node I or amino acid I in mode K, and they are normalized. It's like a probability distribution of displacements. And this factor serves as a weight for those displacements. Lower frequencies have higher weight. So we're moving to the anisotropic network model. Now, so far, the Gaussian network model gave you information on scalar properties, mean square fluctuations, cross correlations, etc. We never talked about what the motion is in 3D because the Gaussian network model doesn't contain that information. By definition, the motions are kind of isotropic. But in the case of an anisotropic network model, ANM, now we get more information from the same data. And as a result, when you extract more information, it starts to be less accurate also, because now we are really trying to filter out a lot of information, which is in 3 dimensions. So we need to be careful. a given structure, again, turned into a network model. The anisotropic network model is going to give you the motions in mode 1, for example, how ProDy interface will give you this type of images showing how different portions of the molecule move in mode 1. So the mode 1 is such an extension. In mode 2, you may see some kind of torsion of the two domains, if you wish. And then if you go to higher modes, you're going to observe much more localized events. So each mode itself, in the mode space, you can think of each mode as a journey in a harmonic, within a harmonic surface. In the case of mode 1, it's relatively smooth. As you go to higher modes, it's very steep. So for a given energy, increment, here in mode 1, you undergo a large displacement. In mode 20, you undergo a small displacement, okay? So that is what each mode represents. Each mode is a collective motion of the entire molecule along the mode coordinate. It's the curvature will be given by the eigenvalue, and we usually organize them in this ascending order. The modes, the first mode corresponding to the most cooperative, lowest frequency, or softest mode, okay? And here's another visualization now. If you look at this HIV reverse transcriptase, for example, an enzyme, here's what mode 1 gives you as the collective motion. Here's what mode 2, mode 3, mode 4, etc. And you can see that, you know, in the first mode, you have a collective movement of the, this is the fingers relative to the thumb and RNase H subdomains. Now you have in ANM2, in second mode, you have a slightly more localized motions. And as you go to higher modes, you have smaller and smaller partitions of the molecule that move concertedly. And the most energetically favorable motion would be the slowest one, okay? Mathematically, it's exactly the same thing as the Gaussian network model. We have a harmonic potential. So this looks complicated, but it just tells you that all internode distances within a cutoff distance are included in this summation. So if they are farther than a certain cutoff distance, RC, the pairs of nodes are not included. The major difference is that now, instead of a scalar here, we consider this as a vector, and we take its derivative after replacing Rij by xij squared plus yij squared plus zij squared, square root of it. Now, when you make these substitutions and you take the second derivatives to construct the counterpart of the Hessian, you get a 3n by 3n matrix composed of 3 by 3 super elements. And each super element here, for example, is has a concise form purely dependent on the interresidue distances in the equilibrium structure that you examine. 0 is the equilibrium. So you can calculate the Hessian matrix. Now you have a 3n by 3n matrix. Now you have the x, y, z fluctuations, not only r, you know, the overall size. And then you can decompose the Hessian as well in the same way. Now the ANM covariance is going to be, if you wish, it is similar in form to the GNM covariance matrix, but each element is a super element actually, composed of 3 by 3 matrices, looking at correlations between X, Y, Z components of different amino acids. So it's a more complicated, let's say, covariance. The diagonal elements actually, if you sum them up, they will be square them up and sum them up. They will be identical to those of the GNM, but now we have more details. Okay. Now, why do we care? Because what we can do using the ANM is to predict what the structural changes are going to be. For example, we can start with from experiments for adenate kinase, we know the open and closed forms. The closed form is the form that binds the substrate. Suppose we don't know the closed form, and we start with the open form. By doing an ANM analysis, we can show that we can predict that structure actually as the second most probable mode of motion. So the enzyme itself possesses the property to reconfigure, to change its conformation into a compact form. So it is already predisposed to undergo this motion. And then when a substrate is around, so this lid, if you wish, these domains, subdomains are going to close down. Same thing for hemoglobin here, the R and T conformations. And you start from the T and you move it along the first or second mode, I don't remember, but you get exactly the R conformation. So in other words, suppose you have a given enzyme and you would like to design a drug or see how it interacts with substrate. It is the enzyme, the protein is going to accommodate protein-protein interactions through the conformational flexibilities it has access to. And those flexibility are nothing else than the modes that we predict. Okay, so theoretically, you can predict the experimental data. And in some cases, the change is really subtle here. For example, you have the bound and unbound forms. You see the bound form is the red one. Unbound is slightly different, but starting from the unbound red conformation, we can exactly predict the blue conformation bound, which optimally coordinates the ligand here. Now, for example, if you consider a large allosteric machine, GNM, and if you consider, for example, to make things easier, let's consider its two subunits only. Actually, these are all heptamers. A heptameric cycle, another ring, another heptameric ring. And what happens is that when there is ATP binding to this form, it undergoes a transition into this more extended form, or vice versa. So there is a transition between the ATP bound and ATP free forms. After the release of the ATP, this extended form of the two subunits, magnified here, converts into a more compact form. Okay, so these are different representations. Now we know from experiments, for example, for this pair, you have the transition between this extended and compact form. Now, if you do computationally, you explore what are the motions in mode 1, mode 2, etc. There's a set of 3n-6 modes, and you would like to see how those modes, which is, you know, an overall vector, 3n-dimensional vector describing the displacement of individual amino acids, how this 3n-dimensional vector compares with the experimentally observed deformation between the two forms. So you can compare that. You can look at the correlation cosine between each of the modes and the deformation vector. And what is surprising is to see that the mode 1, which is the most cooperative motion, gives a correlation of 0.81 with the deformation vector. So for each mode, you can plot the correlation cosine. That is a very high value, showing that, what the Groyan machine is doing is just, sliding along its softest mode in order to undergo those conformational transitions between ATP bound and ATP free forms. But then we thought it is a better thing to compare not only two endpoints, but ensembles. So that was the PhD thesis of a former PhD student, Ivet Bahar. And instead of considering two endpoints for a structure, we have in the PDB multiple structures that have been resolved for a given protein. So we take the ensemble of structures now. And for example, for HIV reverse transcriptase, you have open forms, closed form, DNA bound form, RNA bound form, etc. Now, we were also very much inspired by a paper here in Science published in, I don't remember the year, probably the same year as we published ours, our original PNAS article. So in this paper, they were showing how ubiquitin samples an ensemble of conformations, and then when they have the crystal structures resolved for different ubiquitin molecules complex with other proteins, there is such an ensemble of structure. There is a variation, but the same variation is also observed in solution under NMR, exactly the same type of, variability. Then we said, okay, then NMR in solution actually gives us the conformational variability. And how about calculating the conformational variability for the ensemble and comparing with what has been experimentally resolved to see whether we can really predict ensembles. So there are structural changes like in ubiquitin that accompany substrate binding. By the way, ubiquitin became also the icon of ProDy because we were really very much inspired by that work. And then we can consider structural changes induced by drug binding. For example, P38 is a very important kinase drug target, and at that time there were 182 structures in complex with P38 inhibitors, and all of them with slightly different conformations. Can we predict those conformational fluctuations by the ANM? That's the question. Or let's say chaperonin, as you've seen, it undergoes an allosteric cycle. So there is a succession of conformers that are visited. Can we predict them by ANM? Now, so we do an ensemble analysis, and James is going to show you more details. But the idea is, suppose you have an ensemble of protein structures for one given protein, your target protein. This could be NMR models, this could be X-ray structures resolved in the presence of different drugs or allosteric cycle stages. And it could even be snapshots from your microdynamic simulations. Now, you take this ensemble, you do a principal component analysis to see what the experimental set tells you about the accessible conformational space. Okay, so you look at the major variations in between NMR models, or the functional changes in conformation, or the principal motion sampled in simulations for those three cases. So in the ensemble analysis, you just use experimental data to obtain information on principal conformational changes. But then you compare these principal changes with those from ANM analysis. So for example, you can superimpose aligned structures, and you can evaluate the covariance matrix by principal component analysis and decompose it into a series of modes of covariance, the principal motions, and then you can compare them with the mean square fluctuations predicted by ANM or the covariance matrix predicted by the ANM. Now, note that ANM is based on a single representative structure. Principal component analysis of experimental data is based on an ensemble of structures. So we originally did that for validating ANM predictions, showing that they do provide a good representation of experimentally detected ensemble. But it is even more important when you have one single structure and you would like to predict what alternative conformations are accessible. So we're going to compare theory and experiments. Here are a few examples. So this is an enzyme where, or a portion of an enzyme, where we have done compared multiple structures and got the principal motions from experiments shown in purple, and ANM based on ANM single structure shown in green, and in many examples. And you can see how those arrows obtained from the PCA of ensembles of resolved structures and ANM of a single structure from the ensemble, they show pretty good agreement. That led to an article then in 2009 emphasizing how the structural changes that are observed to be induced upon inhibitor binding are nothing else than the motions intrinsically favored by the molecule, even before substrate binding, drug binding. So this is another way of showing. You have the HIV reverse transcriptase. PC1 is going to give you this type of movements based on the covariance matrix from the ensemble. And then you can compare, you know, what is experimentally observed and what is theoretically predicted. And you can see that experiments show a variation of conformations at the thumb and fingers, and theory also gives you this type of flexibility here. Okay, so we've done for many cases, and in some cases, you know, PC1 and ANM1 agree very well. In some other cases, PC1, first mode from principal component analysis, and third mode from ANM would agree. There may be a change in the orders, but this tells us that the structure in the energy landscape, conformational energy landscape, has access to pre-existing paths. You've heard the concept of pre-existing states. We say that there are pre-existing paths, not necessarily states, and those paths may be conducing to new substates. But those paths, which are nothing else than the directions along the normal modes, pre-exist and enable functional mechanisms. So those are, for example, if you have a protein sitting here and you do ANM analysis, maybe you're going to predict a motion along this direction or that direction, and these might be very close to existing substates, as we have seen now in hundreds of applications. So ProDy, overall, this is a summary. The good news is that you also can start with a sequence and retrieve from the PDB all the structures, and you're going to see in the hands-on all the structures that have this sequence within a certain sequence homology threshold, sequence identity threshold that you can define. Now you can superimpose them. You have an ensemble of structures, or you can do the ANM analysis, compare them, etc. There are many steps, but the idea is that overall, when you start sampling the conformations using ANM, they may be much more broadly distributed than those by MD, because now you have large-scale motions, especially for large systems. ProDy interface has a very detailed tutorial page, which you can use even for learning everything beyond what's going to be presented in this short workshop. And here are the references. This is the original ProDy paper in 2011. Then we got a 2014 version where we emphasized the sequence dependence also. And we wanted to show how sequence evolution affects structural dynamics. So we do think that actually sequence doesn't only define the identity of amino acid or structure, but also exerts a pressure on the dynamics such that the sequence amino acid that's being selected should have a functional motion. Then we had a new version in 2021, and recently James wrote even another review emphasizing its application to cryo-EM. And this is the other dynamics portal that I mentioned. And finally, I would like to acknowledge the two, Ahmet, who was the initiator, currently a senior software engineer working for Google, and Anindita, she made major contributions in the 2014 version. In addition to a few lab members, Ying also was one of the leaders in sequence-based analysis. And then more recently, Honchan Lee, John Zhang, and James Krieger